All right, so let's talk a little bit more about the role of cannabinoid system in immune health. A few words about myself. I am an associate professor of pharmacogenomics with NC State University. Originally come from a small ethnic community up in the mountains in eastern Ukraine. I received my PhD in plant biology from Rutgers University and also an extensive training in nutrition and metabolism from NIH Botanical Center. I am professor affiliated with Catawba College. I consult for standard process, and I don't have any other financial relationships to disclose. So with all this out of the way, let's focus a little bit on today's objectives. So what we would like to do in this talk is to review the endocannabinoid system and its connection to immune health, different levels of regulation of endocannabinoid systems, different levels of signaling, and how they interact with the environment, how they help to interact us with the environment, and also different types of supplementation, for example, botanical supplementations that we can use in order to help those interactions. If you talk about endocannabinoid system, you have to realize it's a very complex and at the same time very secretive system. So in a way, it's the last physiological system so far that we discovered about 30 years ago. And the reason for its complexity is because there are 3 layers of independent regulation within the ECS, that's how I'll abbreviate it. So the first layer is endogenous ligands, right? These are endocannabinoid molecules which our bodies are producing on our own, and they actually acting like a signal molecule, similar to hormones, to regulate different functions. These signals are being pursued, but a second layer of regulation, which are cannabinoid receptors. Obviously, everybody knows about the classical 2 cannabinoid receptors, and I'll talk a little bit more about it later on. But nowadays, we are probably looking at the group of 5 to 6 different receptors, some of them not being specifically cannabinoid receptors, which are known to interact with the endocannabinoid molecules. And finally, once the signal is released, recognized, and processed, right, it has to be terminated. So we have this third layer of organization, which are the enzymes that specifically recognize endocannabinoid molecules, and they degrade them. So basically, they stop the signal from propagating. So this complexity, it's very complex in a way that it happens not only in every single cell of our bodies, it happens different in different types of cells in our bodies. And that's what we will try to talk a little bit about today. So the first thing first, endocannabinoids, right? Here you can see the 2 major examples. And the one on the left, anandamide, probably everybody is very well familiar with because this is the molecule that made cannabinoids famous, right? This is the molecule which we release as a part of our reward system. It's usually produced on demand in large quantities for a very short period of time. And for example, it's responsible for running bliss for those of you who run and know the feeling. After a long run, you know, that kind of little feeling of happiness and euphoria that you get. That's what anandamide is responsible for. Because of that specificity, this molecule is more attracted to CB1 type of the receptors, which are found in our central neuron system, and I'll talk a little bit more about it again down a few slides. While the molecule on the right, 2-AG, 2-arachidonadiylglycerol, is actually the workhorse of cannabinoids, right? This is the one that's produced in large amounts. This is the one that's produced all the time in many different cells, in many different tissues in our bodies. It has a moderate affinity for both CB1 and CB2 receptors, and this is the one that does most of the work, right? So the person, the molecule on the right is the one who's doing the true work, right? The molecule on the left is the one who's having fun. As always, the fun one is, everybody knows about, and the hard worker remains silent. The ways that they are synthesized, it's very interesting, and this is the first connection between the endocannabinoid system and the immune system in our bodies. They both start from saturated fatty acids, which we receive as a part of our diet or we synthesize in our bodies. And the graph on your slide is a little bit too small, right? So I'll break it into sections on the subsequent slides so we can go further. So for now, if you focus on the center part, right? You will see the saturated fatty acids being converted into the unsaturated fatty acid by introduction of double bonds into these backbones. And this pathway, which is common for the biosynthesis both of omega-3 and omega-6 fatty acids, lacks critical two enzymes in mammal systems, right? So if you're an animal, you cannot synthesize linoleic and alpha-linolenic fatty acids. That's why you have to obtain them from diets. That's why they're essential. Once you obtain alpha-linolenic acid, you can actually keep bending the fatty acid structure, right? You basically keep introducing the double bonds, and you lead to a series of fatty acid lipid mediators, which end up with synthesis of EPA and DHA, two classical omega acids, omega-3 fatty acids that everybody knows are good for their health, right? The same pretty much sequence, but with a different structure of metabolite, leads to the synthesis of gamma-linoleic, digoma-gamma-linoleic, and finally arachidonic acid, right? These are the classical omega-6 acids which your body makes. And obviously, a lot of us know that both the homogamma-linoleic acid and arachidonic acids are two fatty acids which sit in the membranes of the cells, and they serve as a precursors for prostaglandin and leukotrienes, the lipid mediators that initiate and propagate inflammatory signals in our bodies. What we often don't realize is that the same arachidonic acid serves as a starting point in synthesis of endocannabinoids, right? So if you look at the core structure of arachidonic acid and core structure of endocannabinoid acid, they're pretty much the same, right? With a small structural difference at the end of the molecule. So this is like one of the easiest and one of the clearest evidence for the tight connection between endocannabinoid system from one hand and immune system from another hand. You see how basically using the same structure of metabolite can lead you to the induction of inflammation, which is driven by arachidonic acid metabolites, prostaglandins are the most famous ones, and slow down and suppression of immune system when you deal with endocannabinoids. And the reason for it is this, right? So here on the slide, I'm basically showing you the structures of lipid metabolites, which are derived from both omega-6 and omega-3 fatty acids using a different enzyme system, right? So this can be your classical cyclooxygenases, lipooxygenases, and cytochrome P50 enzymes. And starting from arachidonic acid, EPA, and DHA, your body produces a variety of lipid mediators, right? Which are used either specifically to drive and promote the inflammation process in the body, or to resolve it once the inflammation is established, right? And just on the left, you see a structure of endocannabinoid, and you see how similar the core structure is, right? So basically, you know, the division is a little bit artificial, and in a way, we are talking about the same class of lipid mediators, right? It's just that they are performing different functions. They either drive immune system, they resolve the inflammation, or they put brakes on the inflammation, and that's exactly what we will talk about. Classical CB1 and CB2 receptors that recognize endocannabinoids, they are conserved in the core region where the recognition happens, right? You see there's anywhere between 40 to 70% homology, and they're a little bit more variable in their flexible regions, which sit outside of the recognition center. These G-coupled receptors, basically they transmit fat-soluble signals inside the cells, where other type of messengers do the actual work. The reason why we define CB1 and CB2 receptors so greatly, and the reason why we pay so much attention to them, is the fact that signaling through CB1 and CB2 is very similar, right? And the tight regulation of both CB1 and CB2 receptors is achieved by spatial and temporal distribution of those receptors. The classical view that everybody knows about is CB1 is mostly restricted to the central nervous system, where it is involved in many different processes like neuronal regulation, memory, cognition, emotion, so our emotional well-being, regulation of appetite and metabolism from a central point of view, and obviously our reward, right? That's why CB1 receptor activation is associated with happiness. The CB2 receptors, because they sit more to the periphery of our bodies, right? So they are located in large amounts in gastrointestinal system, our immune cells, and also our skin and peripheral nervous system, right? So basically, it's the endings of the nerves that give all the perception, including the perception of pain in our bodies. These receptors are more responding to regulation of gastrointestinal health. They are responding to regulation of our immune system, and they are also activation of those receptors leads to decreased inflammation, decreased bone loss, and decreased neuropathic pain. One thing you have to remember, right, that the division is a very gray area. So every single cell in our bodies have both type of receptors present in it. It's just the density of the receptors that it's different. So there is still plenty of CB2 receptors in your brain. There is plenty of CB1 receptors in your gut. Yet the density of those receptors in these systems will be very different. CB1 will be predominantly restricted to CNS, while CB2 will be predominantly restricted to your gut and your immune cells. Looking back evolutionary, it's very interesting to see how ECS system emerge and what it does in our bodies. If you look at the tree of life, going all the way back to one of the first and major splits, which happened about 700 million years ago, we know that ECS system predates that split because it's found in exactly the same functional state and basically including all the same components on both major branches of the tree. So it exists for about 700 million years, at least, right? And it most probably developed further, right, and was supported through all this long period of time by the fact that the life exited the water, right? So we exited the water environment, which is very restrictive in a way that the environment stays pretty much the same all the time. So the changes in temperature, the changes in humidity, obviously, light intensity, are very, very small. There's some difference between day and night, but for the most part, water, large water amount, is a very stable environment. So we don't deal with so many environmental clues that come into our bodies. The moment the life exited the water system and went to the earth, right, you have drastic changes in atmospheric gases. You have changes in atmospheric expression. You have changes in light intensity, UV light exposure, millions and millions of additional stimuli which never actually enters the water system now start bombarding our bodies, right? And ECS system developed, or our bodies developed to use ECS system as a brake for all these signals. So basically, whenever you are overloaded with environmental stimuli, your ECS slowly putting brakes on the activation of your system to make sure that the body can function in the environment properly. One interesting exception for it is insect, and specifically higher insects like spiders, which lost ECS system completely, right? So they don't have a functional ECS. We don't know really why it happens in insects. We don't really know what other system compensates for that loss, but it's an interesting fact that I wanted to share in this presentation. Another thing which connects us, to all other animal is the fact that ECS system functions a little bit different in every single animal. So, for example, if you're looking at human versus dog versus cat and their response to botanical cannabinoids, so in this case, the cannabinoids which come from hemp plants, you will see that the pharmacokinetic profiles for these are very, very different. Dogs respond pretty much the same way that humans do, while cats hardly absorb any cannabinoids into their system. Another level of complexity to this is the response of the receptors as they recognize cannabinoids. So, for example, if you look at the left side of the slide, right, you will see 3 receptor binding assays with human, rat, or dog CB2 receptors, right? And if you look at the curves, they look very much the same, right? So receptor bindings, the recognition of cannabinoid molecules by the receptor happens exactly the same way. Yet, if you look at the panel on the right and you specifically look at human versus dog, you will see that the activation of receptor is completely different. The same levels of binding achieve high activation in human and hardly any activation in a dog, right? So data like this also puts a little constraints of our animal models, because we don't really know how much the data that we get from animal studies translate directly to humans. So hopefully with all this complexity, you learn one thing, and that's that ECS system is a silent system. We don't really know that it's functioning in our bodies, right? If it's doing whatever it's supposed to do. And it's a homeostatic system in a way that it keeps all checks and balances in a proper place. It starts working from the early embryology stage, and it involved in all types of physiological regulation, including neuroplasticity, immunity, inflammation, apoptosis, pain, memory, hunger, and metabolism, right? With such a big involvement in the system, right? Once we discovered all these things, a lot of scientists got excited because they thought, okay, if we know all this, right? If we have a druggable target, we have the cannabinoid receptors, which are specifically responsible for certain effects. Let's try to target them and develop drugs which help us to mitigate a lot of conditions, right? And here's an example of one. We know that obese people or obese subjects with increased cannabinoid tone rate have a chronic activation of CB1 receptor. So it's very interesting to develop a molecule that specifically binds to CB1 receptor and shuts it down, right? So it reduces the activity of CB1 receptor. And several pharmaceutical companies were chasing molecules like this. An example of one of them is rimonabant, which is a CB1 receptor antagonist. It does miracle for the obese patients, right? It helps you curb your appetite. It helps you lose weight very effectively. However, again, remember we were talking about the fact that even though CB1 receptors are restricted for the most part to the CNS, it's found everywhere else in the body. Because of this complexity, right, hitting just specifically CB1 receptor leads to a lot of adverse effects, which are associated with long-term treatment with CB1 antagonists. And what we realized very quickly, I mean, the drug was approved in Europe, right? And we realized very quickly that there is so much side effects, especially connected to psychological and emotional well-being of a person, I mean, up to the point of suicidal thoughts, that within months after being released on the market, the drug was removed from the market and actually never approved in the United States. So with that, with pharmacological targeting of the receptor being out of option, so to speak, the corrective modulation of the receptors, so basically putting selective pressure either on suppressing CB1 or activating of CB2 in order to achieve the desired outcomes, right? Decrease food intake, improve metabolism, and at the same time suppress inflammation, can be achieved by partial selective modulation of those receptors. One of the most famous way of doing that, right, is hemp. And this plant is responsible for giving the name to the endocannabinoid system, because the bioactive principles in hemp, the cannabinoids, were the first molecules which were used to discover the system. You know, in the past, obviously, we did it for pleasure, for religious and medicinal purposes. Now we do use cannabinoids for many, many different applications, I mean, past the historical use. And the reason we use cannabinoids for so many different applications is because of its inherent diversity within the hemp plant. Right, so here I give you just a brief example of the biosynthesis of cannabinoids within the hemp molecule, and you will see that once you start with a single precursor product like aminogyrolic acid, right, you only need 2 different enzymes to perform several steps of reactions to end up with a variety of molecules like CBG, THC, CBD, and CBC, right? The major cannabinoids present in a hemp plant. One thing you have to remember, cannabinoids are always present in plant in their acid forms. So you see there is a carboxy group attached to the molecule, and it's being propagated as the molecule is being modified, right? But it is only when the acid form is removed that the cannabinoid molecule becomes active, or at least activated. Recent studies also suggest that acid forms may have some different effects than the native forms, but we don't have enough data to either support or negate it yet. It's very easy to measure, so I mean, you can have a simple HPLC method which decarboxylizes all acid and can quantify exactly the profiles that you have. And several questions that we often ask when we talk about the hemp plants. The first one is, do I care which part of the hemp plant is being used to produce hemp supplementation products? Because the hemp oils, which are rich in cannabinoids, can be pressed from hemp flowers, they can be pressed from hemp leaves or hemp stems, right? And the short answer is no, there's really not much difference, with the exception of root tissues, right? Because root tissues of hemp don't contain much of cannabinoids. Does the profile matter? Yes, it does. And the reason for it is here on the slide. If you look at the relative potency of different cannabinoid molecules, so for example, delta-9 THC, the famous THC that everybody knows about, it's about 1,000 times more potent than CBD in activating the same target receptors, right? So depending on the profile of your hemp oil and depending on the full spectrum of the cannabinoids which are found in your specific cultivar, grown in a specific location, in specific environmental conditions, the potency of your product can be quite different. Should I care about THC and CBD ratio? It's another question that gets often asked, and the answer is yes. You have to remember one thing. The purified THC and CBDs are drugs according to current FDA regulations, right? So they cannot be used as dietary supplementation products. They are approved for dealing with anorexia and nausea associated with HIV and multiple sclerosis. And CBD, for the first time was approved as a drug to deal with Lennox-Gastaut and Dravet seizures in young children. Because of these approvals, right, these molecules cannot be used for dietary supplementations anymore. Should I care about the terpene profile? This is an interesting question because, I mean, I can both say yes and no to it. Terpenes come in a play when you deal with full spectrum hemp oils, and terpenes play a big role in your emotional response to these products. They carry different types of hydrocarbons, which are perceived by our olfactory and our taste system to have certain physiological outcomes, whether they're stimulative or sedative or refreshing or soothing and comforting, right? And terpenes are not unique to hemp. They're associated with a lot of different plant products. So here you see the example, for example, myrcene, which is found in mango, or bisabolol, the soothing effect of flowers. All these molecules are not unique to hemp, right? Yet the terpene profiles are different for full spectrum hemp oils. So every oil will smell different, every oil will taste different, and it will modify your emotional perception of hemp oil supplementation, right? So think about it as a secondary level that can make your patient or yourself respond better to a particular intervention. Finally, there is a big misunderstanding, so to speak, and that's why a lot of information which is out there on internet, it's often very confusing because the botanical names which stand behind the hemp plants and the street names that we basically tend to associate to these plants when we use them in dispensaries or grow ourselves in the states where you're allowed to grow these plants, there's a very big confusion. Basically, the classical botanical way of thinking about it, right, is having two species, Cannabis sativa and Cannabis indica, and most of the agricultural strain of hemps originally come from the sativa hemp, which is being used today to produce hemp oils. However, a lot of indica plants, which are used for the production of medicinal marijuana and recreational marijuana, actually originate also from Cannabis sativa plant, but from Afghanistan, which is different from the classical Cannabis indica species. So when you talk about people on the street, when you talk about to people in dispensaries, when you go to the internet, you will see the sativa and indica names being interchanged, and often they mean different things in a different products. So something to be careful about. Finally, let's talk a little bit about the direct connection of the ECS system to the inflammation, right? And specifically both to the innate immunity, your rapid response to infection, and your adaptive immunity, right? So your immune memory that remembers the fact that you were exposed to the pathogen or to the infection in the past, and helps you to launch faster and more strict response to a similar infection in the future. To give you the easiest analogy, right, to think about the ECS system, think about the classical fight or flight response that we have when we deal with adrenal hormones, right? So the adrenal system response, which basically is designed to protect our bodies and our well-being in a case of immediate danger, right? So you have only two options. You can either run away from whatever is causing the disturbance in your environment, or you have to fight it because there is nothing to do, right? But either way, you have a chance to survive. ECS does exactly the same thing. It's our fight or flight response, but to chronic stress. Basically, it works in every single cell in our bodies, it works in every single tissue in our bodies, and its major job is to put brakes on the propagation of the hormonal signals in our, or neurotransmitter messages in our bodies. So every time that the environment around us disturbs, right, any kind of visual, auditory, olfactory signals that we get into our bodies, they are translated into either neurotransmitter signals or hormonal messages, right, which are being propagated in the system because the system has to react. However, if these messages are not truncated, if these messages are not stopped, right, you get chronic overactivation of the system, and it's not sustainable. So over time, it basically leads to the exhaustion of the system. So the functional ECS basically puts a brake on it. It suppresses neuronal propagation signals within the neuronal system. That's why it has the ability to lower your pain perception and improve your pain outcomes. It suppresses overactivation of the neurons in central nervous system. That's why it improves your anxiety responses. And it puts brakes on the inflammation propagation cascades, right? So basically, it keeps everyday inflammation within our bodies in check. And this is how it does it, right? So it modulates multiple messages, including the signals. So if we are talking about reactive oxygen species, if we are talking about cytokines, if we are talking about chemokines, right? It directly affects the cell which are producing these messages and basically tells them to shut down this production, to reduce it to the level that immune system becomes functional again. And at the same time, cannabinoids or endocannabinoids have a direct effect on propagation and multiplication of our immune B and T cells, right? So these are the memory cells which keep the memory of the past infections and help you to launch a strong immune responses once this infection happens again. One thing that is critical about inflammation is the fact that inflammation by itself is not our enemy. Every single process in our bodies happens because of inflammation, right? We are physical beings. Obviously, our bodies have very rigid boundaries from our body to the outside environment. The tissues within our bodies are very well defined. And this integrity, right, it's very, very rigid. So whenever you need to change your body, whenever you need to respond to the environment, whenever you need to grow, whenever you need to mature, whenever you need to heal or regenerate the tissues, the only way to do it, right, is to change the tissue by inflaming it. And that means sending a variety of messages there that induce inflammation, increase the blood flow, increase the flow of nutrients in that tissue, and that allows you basically to create pores, so to speak, right? You inflame, so you increase the size of the tissue in addition of the liquids that come to the area of inflammation. You increase all the messengers and you increase all the metabolites needed for the remodeling of the tissues. You do whatever you do, whether it's a repair, regeneration, morphological change, and now you have to resolve the inflammatory process, right? So basically you have to stop it from happening so your tissue can revert back to its new state. So even a healthy growth, any kind of healthy physiological process change in our bodies requires inflammation to be started, maintained, and then resolved. So ECS is critical in the last part, in the resolution of that inflammation. This is one example for it. So if you're talking about immune cells, I mean, in this case of the brains, right? So we're talking about microglia, but exactly the same principle applies to the immune cells in the periphery of our bodies. So for example, macrophages. They typically present in our bodies in their neutral, so-called naive states, when they are basically resting and they are traveling transiently through our tissues, just looking for any kind of external clue to know what they're supposed to do, right? When these clues come in, whether it's a tissue damage, whether it's a pathogen, whether it's a change in the environment, these cells become activated, and usually they get activated into what's called pro-inflammatory or M1 phenotypes, right? Basically saying there's a critical danger at the local level of the tissue or the local level of the cell environment, and they throw a myriad of signals which are pro-inflammatory, right? So we are talking interleukins, we are talking TNF-alpha, we are talking reactive oxygen species, we are talking about nitric oxide, and lipid mediators for the fact, which are derived from the arachidonic acid, and all these molecules tell the cells go into inflame mode, right? Whenever the inflammation achieves what it needs to do, right, whether it's to kill pathogen or to bring nutrients to help the repair and remodeling of the tissues, it needs to be stopped and resolved. And typically, your body is very effective at it, right? And basically switches the activation of pro-inflammatory cells from M1 class to anti-inflammatory cells. The same cells basically morph into the immune cells, which now will stop the inflammation. They stop producing the pro-inflammatory signals and instead secrete the anti-inflammatory signals to basically tell your entire tissues and your entire body to stop driving the inflammatory process forward. Cannabinoids, for a very interesting thing, right? And here we talk about one of the major workhorses, our 2-AG. It activates both CB1 and CB2 receptors, which sit on those cells, and specifically the CB2 receptors, which are found on anti-inflammatory macrophages, and shuts down those responses, right? So basically, your ECS is one of the most important players which leads to continuous suppression and resolution of chronic everyday inflammation in your bodies. This happens everywhere. That happens not only, in central neural system where we were talking about the microglia. Exactly the same process happens in your skin, right? In response to outside injury, in response to increased UV level damage, in response to different parasites which inhabit your skin surfaces. The process is very, very similar, right? You get the activation of the immune cells, which drives the inflammation, which deals with the problem, which needs to be resolved. The resolution of inflammation, again, is handled in part by the activation and propagation of the ECS system. We already talked about the microglia, so I'll probably skip that slide. And there is very big connection, right? These parallels between the brain, these parallels between the skin, these parallels between our gut show us again and again and again exactly the same message. ECS system exists in our bodies to put brakes on the propagation of neuronal and immune stimuli, to keep both of the systems in check, to make sure that the signals which are propagating the defense processes are terminated, and your body has a chance to heal yourself, and prepare for the next environmental clue to respond to. Several examples of additional botanical interventions, right, in addition to hemp, that you can use to modulate those responses. So we'll talk about the classical plant-derived natural compounds, which are present in different plants in different amounts, and their specificity towards CB1 and CB2 receptor. So the first example is the Beta-caryophyllene, which is found in black pepper, Piper nigrum. This compound has a very high affinity for CB2 receptors, right, while it has very little affinity for CB1. So black pepper supplementation or full-spectrum hemp oils, which also contain these compounds, can be used to selectively target the activity of CB2 receptors and decrease peripheral pain, decrease anxiety, and improve immunological outcomes because of the selective activation of CB2. Another example would be Echinacea. This plant contains a group of compounds which are called alkylamides, right? These are also fatty acid-like derivatives with long hydrocarbon molecules, which have selective activity for CB2s, right? So we are looking at about 60 nanomolar potency at CB2 receptor versus about 30-fold less activation of CB1. So you can use Echinacea supplementation in addition to hemp supplementation or in addition of other routines that you use in order to keep the ECS system in check, and by doing that also keeping your immune system in check. Another example, and this is a molecule, in this case, more selective for CB1 receptor activation. It's a molecule called yanganin. It's an alkaloid which is present in kava plants, and kava extract in general can be specifically used to selectively activate CB1 receptors if you need additional activation of CB1. Finally, I believe this is the last example that I have. Falcarinol, it's fatty acid derivative molecule found in carrot plants. Everybody knows about carrots, but this molecule is extremely hydrophobic, right? So it's present in carrots only in small quantities, and it will be concentrated in carrot oil. It's also very likely not to survive any kind of cooking procedure because it's not thermostable. So whenever you cook your carrots, you pretty much effectively destroy these compounds. It's present in carrots in small amounts. So in order to receive a dose which is effective at supplementation and change in the activity of CB1 receptors, you would probably have to eat between 2 and 3 pounds of raw carrots every single day. So it's probably not very easy for a regular person to achieve. However, the molecule itself is very unique in the group of the molecules that I was talking about because it specifically binds and deactivates the CB1 receptor, right? So whenever you have the overactivation of CB1 receptors in a central neuron system, right? So we are talking about emotional well-being, we are talking about anxiety, we are still talking about sleep quality, selective downregulation of CB1, right, and selective non-redundant downregulation of CB1. The molecule basically enters the CB1 receptor and sticks in it. So it inactivates the receptor and you cannot activate it back. It's very desired, so to speak, in order to, again, modulate the activity of the signals between the CB1 and CB2 receptors. So with that description, hopefully I gave you enough of general picture, right? Hopefully I convinced you the fact that ECS and immune system are very closely connected to each other, right? And not so much on a level of the receptors and regulatory pathway, but so much on the level of lipid mediators, both endocannabinoids and major lipid mediators, which are responsible for increase in inflammation, or prostaglandins, are synthesized from the same reactive metabolite, which is arachidonic acid, which sits in our cell membranes. It's involved in every single tissue of our body. It involves into regulation of of our responses to the environment, and specifically, it's involved in the fact that our bodies, by being in the environment, have to deal with the multiplicity of the signals all day long, right? And forget about, stress at work, forget about stress with your family. We are talking about very simple and very basic formula, like increased light, increased sound intensity, something that irritates you all the time, right? All these signals are being propagated within your body. And in order for your body to effectively respond to those signals, these signals have to be terminated. And that's what ECS is good about. It basically puts a brake on the neuronal activation. It puts brakes on your immune activation. So whenever the environmental stimulus comes and challenges you, you basically respond to it, and then you are prepared to respond to the next one. If that balance is destroyed for whatever reason, this is when your ECS tone is dysregulated, and that's when several examples of the dietary supplementation might be able to help.